NYU studying math, background, and physics. And for the past winter and summer, I have been working with a biophysics lab at Princeton, Thomas Gregor's lab, and I've been collaborating with some guys who are now professors at Northwestern and UC Berkeley. If you have any questions at any point, raise your hand or just shout, let me know. But first, I have a few questions for you. First, how many of you are seniors? Juniors? Sophomores? Any freshmen? Okay, cool. And second, and be honest, how many of you have heard about biophysics before even signing up for this class as a field? Okay, that's what I thought. Yeah, so I hadn't actually didn't know that biophysics was a real field until my senior year, and I think part of the reason is because if you ask most people to draw a Venn diagram of sciences, they give you something like this, with chemistry in the middle, and biology and physics on the wings, with biochem and physical chem sort of being the intersection. But I think most of you can see where I'm going with this, that this isn't the real picture of the three sciences, that in fact it looks something more like this. And really what I'm hoping if you get nothing else out of the talk today, I want you to go away with understanding that there's this real feeling among high school students and undergraduates that if you're a biology student, you're a biology student, or if you're a physics student, you're a physics student, that you have to pick one specific field to be in. And for some people that works out great, but it's also important to know that your choices aren't limited. And this is just for the three, physics, biology, and chemistry. If you add computer science, math, the graph becomes far more complex. So the number of subfields at different intersecting points is enormous. And so there's a lot of cool stuff you can do that's sort of outside your typical picture. So if we're going to talk about biophysics, I guess I should first define it. Biophysics, and this is my definition, by the way, so yeah, it's by no means a definitive definition. But to me, biophysics is where you take the subject, the thing of interest in biology, which is life, and you apply physical methods and physical intuitions from physics. I think most people would agree that biology is definitely a messy field. You're both quite literally messy, since you're dissecting and whatnot, but also quantitatively it's messy. Whereas physics is 100% quantitative, you're doing a lot of working with numbers the entire time. Biology is far more sort of qualitative. And so a lot of people, particularly in the past century, have been looking for ways to apply mathematical techniques and models to biology and make it a more sort of legitimate science. And that's would be more biomath. And some of the most exciting research that's being done at Courant is in biomath. Some really cool stuff. But to me, the issue with biomath is that it becomes a little too abstract. You're trying to use very abstract ideas for a science where basically we're used to just looking at stuff that's everyday stuff and trying to figure it out. So applying topology and a lot of this really complex math to biology can be difficult. And that's where biophysics comes in. It sort of bridges the two worlds. It makes biology much more quantitative, but at the same time it allows you to keep your sort of physical intuition of how things should work. Okay, so let's talk a little bit about the history of biophysics. There's quite a surprising amount of history, even though it's a relatively new field. It's probably one of the newest fields. And in particular, the thing I was most surprised to learn about is that, I guess, who might call the first biophysicist was Alan Turing. Have you guys seen The Imitation Game? Have you seen the movie The Imitation Game? No?
Yeah.
Yeah, okay. Yeah, it's good. So Alan Turing, he's known as being the father of computers. In the movie, he is played by Benedict Cumberbatch, the Sherlock guy. And in addition to that, what we're more interested in is that he developed binary programming, this idea that you can store information with ones and zeros. The whole binary thing wasn't his idea to begin with. that has to go back to the pool. But he was the first one to realize that you could store information using ones and zeros. And this is going to be play a lot into genetics, where we talk about whether a gene is on or not. That's sort of 1 or 0. Also, he is the father of artificial intelligence and the idea of wondering what distinguishes life from machines, and which we'll look at a little bit. And then finally, he is one of the first ones to come up with this idea of morphogenesis. At the end of his life, he published a bunch of biology papers. We'll talk about this in a second. The second founder, I guess you would say, of biophysics is John von Neumann. I don't know, has anyone heard of him before? Von Neumann? Okay, he's sort of on parallel with Turing, both great polymaths of the 20th century. And he did everything from founding game theory to what we're more interested in, coming up with models for mathematical self-replication and cellular automata. He is interesting because von Neumann was able to sort of, by about 30 years, predict how we genetically pass on information. He came up with a mathematical model for understanding how we could pass on information. And so he took Turing's ideas and took them a little bit further, and then finally, the guy you're most familiar with is Francis Crick, of Crick and Watson, the DNA guy. And he came up with this idea of a morphogen gradient. Okay, so I've said morphogen twice and gradient once, so I want to talk a little bit about what exactly a morphogen is. A morphogen is a protein, little particle, that floats around in a cell. The most important thing is not knowing the biological roots of it, but instead understanding sort of the big picture. So I'm going to give you, it sounds like it's going to be cheesy, but honestly this is the way I think of morphogens. Have you guys been to those places with the ball pits? Yeah, like where you climb around, they're really unhygienic. Yeah. Okay, so if I took a big bag of those little plastic balls and I dumped them right here, and just dumped them, and if we didn't have any chairs or tables, we would be able to count up the number of balls in each square, and that would sort of define what we're going to call a gradient. This idea that at any given point in this room, there would be a set number of balls, and the majority would be over here. There might be 1 or 2 down there, but there would be some sort of continuous flow. And this is important in early developmental biology, which we're going to talk about more. But just to understand this idea that morphogens, you don't have to understand them biologically, but you should start thinking about them as sort of ways we can pass information. Because if you know that I dumped the bag of balls here, then you can probably guess how many balls are going to end up here and how many are going to end up here. Or if you are given a field, if you're given the room after I've dumped them and you know that there's like 1 ball down there and about 30 over here, you can guess where I've dumped the balls to begin with. So it's a way of sort of reconstructing biologically what happened after the fact. And that's something we're going to explore. Any questions? Okay. So that's all well and good, but what about current research? What's happening now? So here's some of the guiding questions that biophysicists use to sort of guide what the research that they're doing. So first of all, what is life? That's sort of the question of biology. I would argue that we have made very, very little progress on answering this ever since the Greeks asked this. But it's still a fascinating question, and I think if you ask most biologists and biophysicists why they got into the field, they would say that they were fascinated by the question, what is life? Second, how does complex life emerge? This is definitely something that is more answerable, but we certainly don't have an answer, and I would say aren't even that close to having an answer. But again, we're sort of refining our questions. And finally, how does life take discrete information and give a continuous response? And how does life take a continuous stimulus and give a discrete response? Everyone knows what I mean by continuous and discrete. Yeah? No? Okay. So discrete would be integer values, or like a binary system, where in a given system you can have one of 2 values or one of 3 values. You can't have a half value. You can't be in between. You have to be either 0 or 1, or 1, 2, 3. You know, there's a discrete, a given number of values that your function can have. Whereas a continuous function would be something like the number line. From 0 to 1, there's an infinite number of numbers, and that they never break. So I'm sure maybe you have seen, if you have a graph and you have something like this would be discrete. You have values either here, 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 whereas continuous would be something like this, where you can have values anywhere along this line, if that makes sense. So in particular, we're going to be looking at how life takes continuous information, something like a gradient, which has a sort of an infinite number of values it can have, and how it turns how it has a very discrete response. Either it responds with a 1 or how it responds with a 0. So to do this, we're going to look at the sort of the theory, the most important theory in biophysics and in developmental biology, which is a French flag model. It was developed by this guy named Lawrence Wolpert, in 1968, 1969, and was sort of refined by Francis Crick in 1970. So the basic idea is, well, here's the French flag. If we had a box of light bulbs, and each light bulb was given a certain task, it had to read the number of morphogens, or in the example I was giving before, plastic balls at a given point. Each light bulb can have one of 3 responses. You can either have a blue light, a white light, or give off a blue light, a white light, or a red light. And what defines what it does is the number of balls or morphogens it reads. So what you would have is you would end up having something like this. If you had a sort of a rectangular box and you had lots of little light bulbs all around. And if the balls or morphogens started here and sort of flowed outwards, you would end up sort of with essentially the French flag, where if the blue light was triggered by sort of the most balls, and the white light was triggered by a medium number of balls, and the red light was triggered by 0 to, say, a few balls, then what you would end up having is quite literally a French flag, roughly. And so this idea of a defined response pattern is very interesting, and something that we're going to look at in more depth. In particular, we're going to look at the so-called Bicoid gradient, and how it leads to different, sort of a 50-50 split along the fruit fly embryo axis. So this would be, if this is an embryo, and these are all the nuclei, here would be sort of a zoomed up picture where you have each one of these ones is reading the number of morphogens and is having a response. So all these white ones, they've read too low morphogen to turn on. And all these ones, the other colors, they've read a certain amount of morphogen that's high enough for them to start responding. And in particular, when I say that they're turning on or staying off, I mean that they're either expressing a gene or they're not expressing a gene. I'm assuming most of you have studied some DNA and whatnot. The slightly more complex picture that you might or might not have looked at is if you had a strand of DNA coming along, the strand is not always going to be read and copied by mRNA and processed. What decides if it is or isn't is this little thing that acts as sort of a lock. And unless there's a key to fit in it, it's not going to replicate. There's not going to be any DNA replication. So one of the ways we think about morphogens is that they trigger a key to come in here, and this to be unlocked, and then the DNA can be read. So how do we do this? How do we look at cells? How do we study the development of tiny little embryos. Well, in particular, the lab I've been working at uses this new technique called live imaging, or in situ imaging, which is Latin for live and in this very situation that you're looking at. So it's sort of like if you want to study a wild lion, there are a couple of different ways you could go about it. You could either go out to the Sahara and kill one. It would be great because you would have the lion that from the natural habitat, but it would be dead. So you couldn't do anything. You couldn't look at anything, any of its behavioral patterns because it's dead. Or you could go to the zoo, like the Brooklyn Zoo. You could look at lions, and that's great because they're alive. But in this case, they're not in their natural habitat. So you're missing something. The best thing to do would be to videotape or watch the lions in their natural habitat without disturbing them. And that's what this live imaging does. It allows us as researchers to look at the nuclei in its natural environment while it's still alive. So we get the best of both worlds in this case. And I really like this technique because it takes advantage of both biology and physics. It really shows why biophysics is biophysics. In particular, it takes advantage of two things, two-photon microscopy and fluorescent mRNA tech. So I'm going to show you just a little video of what it actually looks like. Or not. Well, I apologize, never mind. Maybe. I don't really know how these projectors work. I apologize. But you can see that the red things are the nuclei themselves, and then the little dots that light up, those are individual nuclei that are turning on, that are turning on. The ones that, the red spots that aren't turning on have stayed on. Okay, so looking in more detail, so the way we capture the images are using two-photon laser microscopy. It's very fancy. I don't pretend to really 100% understand it, but a bunch of the graduate students at the lab I was working at actually built one, which is really cool. And basically the main thing to understand is that you're using very fancy optics and physics to take a laser, very high power laser, and by sort of bouncing it around, you target it at the embryo, which would be right here. And the photons that are emitted from the laser, and photons are just light particles, they hit the embryo at a particular point, and then they bounce off either up or down, depending on where they hit. And then they're collected by these PMTs, which are things that capture the photons and allow us to reconstruct the images. So again, like I said, I definitely don't pretend to understand it, but it's very cool, very complex optics. And so if you're interested in that sort of stuff, building microscopes is a great way to help science and do some cool things. And this is just a picture of it. Wait, so how about fluorescent marking, which is another thing of interest, and one of the things that allows us to take these cool videos. This is not, by no means, is this unique to biophysics. It's used in sort of all bio labs. So if you're ever working in any biology lab, you'd be probably, might be working with something like this. So, like I was talking about earlier with the DNA, mRNA comes and cuts this up open. And you have the individual nucleotides. And so mRNA comes by and makes an exact replica. And that's used to make the proteins. Is that familiar with everybody? Yeah. Sorry. Okay, so what we do is we bathe the fruit fly embryo with all these fluorescent PP7 GFP particles. They're essentially, the GFP is this little like light. It's fluorescent. You can see it with the microscope. And the PP7 fits into RNA. So essentially what we end up having is all these mRNAs that are tagged that you can see with the microscope. But the reason you don't see them normally in the cell is because they're very diffusive. So sort of like when you're coming back from flying, when you fly over, say, Jersey, it's way, you don't see that many lights, but as soon as you hit Manhattan, the whole thing blows up because there's so many lights. This is sort of a similar idea that when the cell just is sort of bathed with these lights, you don't see anything. But when our DNA replication starts, all of them become concentrated in one area in this sort of, in the making of the mRNA, the copy. And then we can see very specifically where this is happening. Which is important because we want to know specifically what genes are being turned on and where these genes are being turned on, where they're being replicated. Because if we can understand where they're being replicated in which genes, then we know which proteins are being turned on. Okay, so using this new data and technique, my collaborator at Northwestern came up with sort of two ideas by messing around with one of these data sets that we ended up getting. And those ideas were that there might be interactions between nuclei and strings. Now, I should clarify, the paradigm, the French flag model, allows for neither of these two things. That's what makes this interesting, is that we're looking at perhaps revising the French flag model, that there's clearly some issues with it, and that these two things are some of the things that we think a correct model should incorporate. So, in particular, when I say interactions between nuclei, I mean that there's a large number of active nuclei that have neighbors that are active, which sort of makes sense. It's like, well, I don't know, maybe not, but yeah, if you guys, when you were signing up for cSplash at school, if one of you chose a certain class, then more likely some of your friends might also choose that class. This is a similar idea that rather than just reading the morphogen and either turning on or staying off, each nuclei checks with its neighbors and says, hey, are you active? Are you active? Because you could think that maybe a nuclei misreads. And if a nuclei misreads and doesn't turn on, that could be an issue. So it makes sense that you might check with others to see specifically have they turned on or have they not. This is one of the preliminary graphs, the data being in black. These other three, I don't know if any of you have taken stats, but they're the null of the data. They're just what we would expect given the French flag model. If the French flag model was exactly the way it was, if the nuclei and the embryo obeyed the French flag model exactly how it was, we would expect something like the data in silver. But instead we don't, we get something that's like the black graph. And so, particularly on 2 and 3, there's more neighbors, or active nuclei with 2 or 3 active neighbors than the French flag model would suggest. Now, like I said, this is just with one data set, so this was a curiosity, and it was something that we wanted to look at more. Also of interest were strings. This is a live image from the imaging technique, and these are all the embryos, all the nuclei, and each one is, we take all the active nuclei, those are the ones in white, and we look at their orientation, whether they're straight up and down, whether they're completely horizontal, whether they're tilted, and we plot that pneumatic, that's what it's called, pneumatic alignment in red. And so, as you can see, sort of right here and right here, it looks like the red lines line up. It sort of looks like the way the nuclei have ordered themselves is stringy-like. And that was also something that is not explained by the French flag model, and it was certainly an interesting topic that we wanted to look at more. So, just before I talk about what we actually did, I wanted because most of this might sound like biology, I want to bring back the physics and talk about bringing our physical intuition into this problem. So I don't know if, maybe just for my hometown, but if you go to the pediatrician, a lot of like the children's doctors, a lot of them have those toys with the wooden rod and the little wooden balls that are attached to it. Yeah, you know, okay. Okay. So yeah, one of the cool things is if you spin one of them, sort of spin it around, some of the other ones also spin. And so the idea is that by changing the orientation of one of the little knobs, or little balls, you can change some of the other ones. Now this is sort of obvious because there's friction, and by pushing down you're creating a torque which moves the other ones. But the main idea is that there's something very comparable to this in statistical mechanics. Now you don't have to understand the equation or any of the real physics, but it's important to sort of think about this problem like we would, like in a way that we can physically understand. And that is that when these particles spin, either up or down, they exert an influence on other particles to spin like the way they do, which is captured by this equation. And so the idea of interacting particles or interacting nuclei isn't as strange as it might sound otherwise, because we see it all over the place, whether it's in statistical mechanics or in the pediatrician's office. So now taking this sort of physical model, we're going to start applying it to the problem we were talking about earlier, in particular looking at for strings and interactions. So our first object was to look at the static picture. At the very end of sort of division, we wanted to look at how do the patterns look at the very end, just like if you took one snapshot. And this is not the sort of the real interesting part, but it allowed us to say definitively whether there seemed to be patterns of interest or there didn't. So first thing we did was we took all the active nuclei, all the nuclei that at some point in cell cycle 14 had turned on, had started expressing some gene, and we looked at the number of active neighbors they had, which is plotted here. And the count is on the vertical axis, which is the percent of active nuclei with that given number of neighbors. So as we compared this for statistical reasons to inactive nuclei and looked at specifically how many active neighbors the inactive nuclei had. And so what you would imagine is if the French flag model was exactly the way it currently is, that there should be no difference between the two. It should just purely be a binary thing. Every nuclei reads the field, reads the number of balls in the area, and either turns on or turns off. But that's not what we were seeing, because as you can see over here, inactive nuclei have far more neighbors with have far more zero active neighbors, whereas the active nuclei have far more 5 and 6. So the picture we're sort of getting is that if you took all the nuclei, or specifically if you took one nuclei, and you look at, for instance, this one, if it's on, then it's far more likely that all of its neighbors, or most of its neighbors, will be on. Whereas if it's not on, then far more of its neighbors are not going to be on. And this is sort of definitely what we were looking for when we were looking for interactions. There's clearly this idea that if we find one that's active, then we can reconstruct whether its neighbors are active or not. And this would suggest that it's more than just a purely binary reading the field and turning on or off, that there's something deeper that we wanted to get at. And this was reflected when we created a simulation. I don't know if any of you are interested in computer science, but it was a lot of fun. We created 1,000 random orderings of how the nuclei could be. So we took the embryo, and it said, say, it had 300 nuclei. And with each simulation, we sort of chose at random whether the nuclei would be, any given nuclei would be on or off. And this created sort of just a random, like we were just shooting randomly what the pattern would be. And as you can see, this is our data, and this is all of the random simulations. And so if you know nothing about statistics, it's still pretty obvious that our data is not like the random simulations. And so one thing we can conclude from this is that if our data is not like how we would expect the data to be, given the French flag model, and particularly if it's so different that there's like a 100 chi-square gap. Chi-square is just a statistical test. If there's a gap that big, then you would imagine that there has to be some other explanation for the phenomenon we're seeing. And this got us really excited. But what about strings? Like the picture I showed you. Well, unfortunately, when we looked at more data sets, we found that strings were just sort of a fluke. It only happened in one. And you can see this, again, this is sort of a more complex thing, but this is our data versus a null of eccentricity alignment. So essentially, if the average nuclei should follow this trend, should sort of shift in a specific sort of random way, we would want our data to be different on the ends here, so that more should be pointing this way than expected, or more are pointing this way than expected, because they should sort of line up together. But that's not what we found at all. It really was just random. And this was confirmed in this graph. We were sort of looking for the data to beat out the null, which it didn't. So we could cross off strings. That's a lot of what science is just looking at specific phenomena that you see once or twice, and then getting rid of some of the ideas and confirming others. Yeah. Okay. Now that was interesting, but that wasn't so particularly interesting, so we wanted to look more at what happens sort of spatio-temporally. As you go through space and as you go through time, what exactly is happening? How are the phenomenon, how is it getting to be the way it is? How is the pattern getting to be the way it is? And so we looked at, again, the nuclei, the number of active neighbors they had, but this time we looked at when they turned on, when the nuclei turned on, whether they, exactly when they turned on, how many active neighbors they had. And again, we came up with 1,000 random orderings, just really cool computer stuff. And then we plotted our data versus this, and again, as you can see, even if you don't know much about statistics, clearly our data is very different on the lower end than on the higher end, which again got us excited because this is sort of exactly what we would be looking for if there were interactions. Yeah, okay, and then also we did some fancy statistical stuff, created cross correlation. We wanted to look at if you have this nuclei here, all of its immediate neighbors are, say, its first shell. All of its people that are closest to it. And again, you can apply this to, like, I don't know, your friends at school or something. That we looked at this nuclei, its first orbital, or its first shell, then its second closest neighbors. Third closest neighbors, etc. And we wanted to look at how much influence does one nuclei have on everything else. And what we were hoping for is that it would sort of exert an influence on the first and second shells. It wouldn't influence everything. Because then it would just be like if one nuclei turned on, all of them would turn on. And that's not what we had seen. And this sort of corroborates what we were hoping for, that really the data is only different from the null just for the first and second orbitals. Yeah, which again was what we were hoping for, and more proof of this idea of maybe there being interactions. Yeah, okay, so one of the questions that you might have is, well, how do we know that it's truly interactions? How do we know that it's not something completely different, like, I don't know, heritage? In the 13th cell cycle, every cell splits so that in the 14th cell cycle, there's the mother cell and one of its daughter cells. And so you could say, well, how do we know it's interactions? How do we know that it's not just completely familial or heritage based? That if a mother turned on, and then its daughter will turn on in the next one. So we looked at that. We wanted to make sure, cover all our bases, that we weren't just getting excited about nothing. And so we sort of came up with some probability distributions on if the mother turned on, how likely is it that the daughter would turn on? And if the mother didn't turn on, how likely is it that the daughter wouldn't turn on? That's what these numbers are. And the one thing that should sort of jump out at you is that they're all pretty much the same. And so this would seem to suggest that it's not heritage-based, that there's no real difference between the probability of if a mother was on, will its daughter be on? If that makes sense. So we were sort of able to rule out this heritage factor simply by just looking at the data and saying whether creating these probability distributions and then looking more specifically at whether they were different from each other or not. Okay, and then in the future, we're looking at another gradient. It's called Eve2. It's not really important, but instead of creating this sort of division between the head and the tail, it's sort of a striping pattern, and it helps it create a difference between the dorsal and ventral sides of the embryo. And also, we're currently in the middle of building a model to explain all this phenomenon and whatnot. Okay, so I also want to talk a little bit about how you guys can get involved in research. I know particularly for juniors and seniors, you have college on your mind. This was sort of unique because the professor came to me, not the other way around. But I had done some work for sort of this high school program in Philadelphia at the Fox Chase Cancer Center, and through them they hooked me up with this guy at Princeton. I think the most important thing is if you're really interested in doing research, whether it's in biophysics or anything else, you want to look for programs that are perhaps specifically targeted for high school kids. There's a lot of programs nearby that are looking for kids who are young but really smart and really interested in what they're doing. And also, you sort of want to put yourself in the position to get lucky, because most of it is really luck. So the more you do, the more chances you have to get lucky and do stuff that you might not otherwise have been able to do. Yeah, that's pretty much. I mean, I'm sorry I had planned this talk to be longer, but sort of rushed through it. Are there any questions? No? Okay. Yeah.
Do you have an example of a program besides the one that you went to?
Not in New York City. I'm from Central Jersey, so. But I'm sure I could find one. Oh, by the way, since cSplash is supposed to be more, not just a one-day event, but sort of a way for you to build a community of people who are interested. Here's my email address, should you have any questions. And if you send me an email, I'd be happy to find out for you and send something back. Whether it's about NYU or biophysics or whatever, I mean, should you have an interest. Yeah, any other questions? Okay, well thank you guys so much again.